Okay. Hello, and welcome to today's CTSI Access Technology Program Seminar. I am Giselle Jacobson, the staff scientist in the Biophysics Instrumentation Core facility, or BIC facility, at the University of Notre Dame. The BIC offers instrumentation dedicated to characterizing biomolecular conformations and interactions, as well as equipment for isolating and purifying macromolecules for subsequent biophysical analysis. We currently have 13 pieces of equipment, which are available for in-person or sample mail-in usage. Some of the instruments that I'd like to highlight that we're not going to be talking about in detail today include our JASCO circular dichroism spectrophotometer, which provides information on molecular structure and its changes for various chiral molecules. We also have a Beckman analytical ultracentrifuge, which measures macromolecular size and shape and studies interactions in solution as well. And then also a microcal isothermal titration calorimeter, which measures the affinity and enthalpy of interactions in solution. So for more information on the instrumentation we have available in the BIC, please contact me, or you can visit our website. And I'll leave information, the contact information in the chat session. So today it is my pleasure to have Dr. Eric Rausch from Sativia here to talk about Biacore surface plasmon resonance, which is a technology available here in the BIC. We recently acquired a Biacore T200 through an NIH S10 grant. Dr. Rausch is a principal application scientist at Sativia. He received his PhD in biochemistry at Duke University and then joined Biacore as an application scientist back in 2000, before Biacore was acquired by GE Healthcare. So he has over 21 years of experience with Biacore SPR. So without further ado, let's welcome Dr. Rausch.
Okay, so today we'll talk about Biacore surface plasmon resonance instruments and what they allow you to do, which is essentially to observe molecular interactions in real time. So we'll start with laying some definitions out, setting the ground, as it were, for this talk. For purposes of today's seminar, we're going to consider biological interactions as real-time binding events where biomolecules, which could include small molecules, associate with and dissociate from other molecules. And Biacore instruments allow you to measure these interactions in real time without use of labels, using the phenomenon called known as surface plasmon resonance, or SPR for short. Couple of important definitions. The analyte is the interaction partner that passes over our sensor chip surface. Ligand is the molecule that we attach to the sensor chip surface, which hopefully will interact with our analyte. If you're coming from a receptor biology background, this terminology is exactly backwards, and I apologize for that. All I can say is that it's been this way for 30 years and it's unlikely to change now. So remember, if I mention ligand later on in the talk, it will be the molecule attached to our sensor chip surface, and analyte will be the molecule that flows over the sensor chip surface and interacts with the attached ligand. So why are we interested in monitoring biological interactions? Well, if we're doing protein research, this can give us significant insight into structure-function relationships. How do these two molecules interact? Which residues or side groups are involved in coordinating the interaction? What happens if we make a change to one of these groups? Does that increase or decrease stability of the interaction? From there, we might go into signaling cascade studies. If molecule A binds to B, can molecule C then still bind to B, or does that block access of molecule C to molecule B? For drug discovery, we're typically looking for molecules that usually will bind proteins that are somehow affected or associated with a disease state, and either to inhibit protein activity, or sometimes to actually enhance protein activity, depending on the effect of the protein on that disease state. With respect to development and quality control, once we've made a biological drug, it can be very important to measure its affinity and kinetic parameters on a batch-to-batch basis to make sure that we're seeing the same behavior in each batch that we make. If we have a significant deviation in one particular batch, that may be cause for concern, allowing us to go back and examine what may have gone on, and if necessary, discard the batch and proceed forward with making a new batch. So how are molecular interactions described? Affinity is a measure of how strong your interaction is. The classic equilibrium dissociation constant, or large KD, essentially that concentration where at steady state, 50% of your reagents are in complex form, as opposed to being free reagents. Kinetics tell you how fast or slow these complexes form decay. Ka, the association rate constant, determines how quickly molecules bind to each other. Small kd, the dissociation rate constant, is a measure of how quickly or slowly these complexes break apart. And we can also describe active concentrations of molecules. And not the difference between an active concentration and a total concentration can sometimes be relatively significant. We may have a significant difference between the amount of material observed on a Kumasi stained gel, is a nice big blue band, and the amount of that material that's actually functional, capable of interacting with target molecules. And it's always good to know how much of a given batch is functional. And we may also be interested in specificity. For a family of molecules, can we find a binding partner that only targets molecule A, not A1, A2, or A3, but just A, in the binding specifically to A and not to any of its related family members? So there are three core components to Biacore instruments. One are our sensor chips, to which you attach a ligand. Two, microfluidics, which are how we get sample to the sensor chip. And three, surface plasmon resonance itself, how we measure whether or not binding events are occurring at the sensor chip surface. Take a look at our sensor chips first. A typical Biacore sensor chip will have a glass support that is optically matched, has the same refractive index as the prism inside the instrument that is focusing light on the sensor chip surface. On top of the glass support, we have a gold layer. It is very thin, only 50 nanometers thick. The gold provides a source of free electrons to amplify the surface plasmon resonance effect. Most of our sensor chips will then have an alkane thiol linker layer on top of the gold to minimize non-specific binding events of biological molecules to gold. And then finally, on top of the alkane thiol linker layer, there is usually a carboxyl methyl dextran surface. The dextran itself is unbranched and fairly inert with respect to biological activity. The carboxyl groups provide you a suitable attachment point through a variety of coupling chemistries, amine, thiol-based, or aldehyde-based, to put your ligand on the chip surface. The overall stability of your chips will be dependent on the molecule you attach to the chip surface. That molecule, the dextran itself, is very robust. I have used sensor chips that were 5 years old and still gotten good activity from them. But once you attach a biological molecule to the surface, the lifetime of that surface is now equivalent to the lifespan of that biological molecule. Microfluidics bring sample to the sensor chip surface. Our sensor chips themselves are planar. The integrated fluidic cartridge, or IFC, forms the various flow cells on the sensor chip. And the flow cell number can vary from 2 to 16, depending on which Biacore instrument that you're using. The Biacore T200 at the Notre Dame core facility has 4 flow cells. Typically, we will use either 1 or 2 flow cells as reference, and 1 to 3 flow cells as active surfaces where we have attached ligands of interest. Why do we need microfluidics? Well, it gives us a number of advantages. It reduces sample volume. Typical samples will be on the order of 50 to 250 microliters. Concentrations will be in the range of the interaction affinity that you're trying to measure. So if the KD is nanomolar, you use nanomolar concentrations of the analyte. If the KD is micromolar, you'll use micromolar concentrations of the analyte for your samples. Associated with the low sample volume, there's also relatively small dead volume. If you're doing even a 250 microliter injection, you probably will not need to put more than 300 microliters total in the tube where the sample will be stored. Using microfluidics lets us put multiple channels on our center chip surfaces. It also allows us to do inline reference subtraction so that we can subtract signal from our reference surface directly from signal from our active surface to give us a reference subtracted data that will be easier to analyze and work with. We're trying to minimize dispersion and dilution of our samples so that the sample we inject is at the same concentration when it reaches the center chip surface as it was in the tube before it was picked up. Microfluidics also allow us to have relatively fast temperature equilibration times. Our center chip might be at 37 degrees C. Our sample compartment might be at 10 degrees C. By the time the sample gets to the center chip surface, through a combination of relatively small volume and passing through a relatively long, narrow IFC directly seated on top of a Peltier device, by the time sample gets to the center chip surface, it will have equilibrated to the temperature of the center chip. Now, for the $64 question, what exactly is surface plasmon resonance? When we shine plane polarized light at the interface between center chip and buffer, if the light is above a certain critical angle, we have total internal reflection of the light. Note its light is being reflected by hitting the buffer, which has a different refractive index than the glass of the center chip. When we have total internal reflection, one specific angle of light will lose energy as a function of the refractive index of the reflecting medium. If this refractive index changes, then if the refractive index changes, then this angle of light that loses energy also changes. So we can monitor binding events as a function of change of refractive index at the center chip surface. We've got the SPR angle. As we inject a binding partner, if it binds, angle of light loses energy, or the angle of light that loses energy shifts. We measure that shift with our detector and can translate this directly into mass changes due to binding events on the center chip surface. So typical data from a Biacore experiment will look like this. We have a baseline signal where we are flowing buffer over the center chip surface. We inject analyte. Hopefully, we see a binding event. At the end of that sample injection, we switch back to running buffer over the surface. Biacore instruments are flow-based instruments. We will always be flowing sample or buffer over a surface unless we specifically tell the instrument to stop. In the association and dissociation phases, we have information about K off. Additionally, in the association phase, we will have information about K on. If we can measure K on and K off, we can calculate affinity. If we are doing concentration assays, then we may look at a specific point in time for all of our samples, use that to generate a standard curve and compare unknown samples to our standard curve. So how sensitive is a Biacore surface plasmon resonance instrument? We can measure interactions where the analyte to ligand mass ratio is low as 1 to 1,000. So that could be a 100 Dalton drug or fragment of a drug binding to 100 kilodalton protein. Here we can see as an example, the 100 Dalton analyte and the 50 kilodalton protein. If we put 5,000 resonance units on the sensor chip surface, we would see up to 10 resonance units of this analyte bind to the ligand. Given that the noise level on the Biacore T200 is less than 0.2 resonance units, there is more than enough signal noise to accurately measure K on, K off, and affinity for this interaction. Just an additional note, 1 resonance unit is equal to 1 picogram of protein on the sensor chip surface. We are collecting data in seconds. Biacore T200 can measure at 1 hertz, 1 point per second, or 10 hertz, 10 points per second. So we can measure K on up to 10 to the 6th per molar per second, K off down to 10 to the -1 per second. If events are significantly faster than that, we may get what you would describe as a square wave sensorgram, baseline, automatic jump up, saturate, or to a steady state binding level, and then automatic jump back down to baseline at the end of the injection. Under those conditions, you will not get kinetic measurements, but you can still measure affinity as a function of steady state binding levels plotted versus concentration. With respect to the range of affinities, Biacore instruments have reported picomolar interactions, typically antibody-antigen, all the way to millimolar interactions, where you may be doing an initial drug library screen against a target. So we can cover 9 orders of magnitude of affinity. It's fairly impressive. So benefits of using Biacore SPR. It is a label-free system. We do not need to tag our molecules with a radio label or a fluorescent group in order to see binding events occur. This reduces the amount of time that we have to use to prepare samples and minimizes the chance of an artifact due to the addition of for example, a relatively bulky hydrophobic group that has fluorescent properties. It is a real-time event. We can see binding as it happens, so we can measure interactions that may be relatively slow or extremely fast because we are catching it from beginning to end. It is a contact-free approach, not invasive. We can work with fairly complex sample mixtures, samples that may be resistant to letting light pass through it, because for SPR to work, light only needs to be reflected. It does not have to pass through the sample. I had a colleague many years ago, a research group was interested in studying peanut allergens. So they set up a test with Reese's peanut butter cups. Took them, homogenized them, filtered them, so you had a nice chocolatey, peanut buttery extract, passed it over a chip coated with peanut allergen antibodies. And yes, as you might expect, there are peanut allergens in Reese's peanut butter cups. Not really a surprise there, but that's just an example of how complex a mixture can be to still give you acceptable Biacore results. All right, so before I jump into applications, are there any questions?
Yes, we do have a question. Okay, go ahead.
I see it. How do you deal with non-specific binding? That's a very good question. We have a variety of sensor chip chemistries that you can use that have more or less carboxyl groups on them. In general, if your non-specific binding is electrostatically driven, then using a sensor chip with reduced carboxylation will reduce non-specific binding. If it's a more hydrophobic effect, then you may want to go to adding detergent to your running buffer or some other modification to reduce the hydrophobic interaction that may be going on. And those do tend to be the 2 major drivers of non-specific binding. Third possibility is that you have something that specifically binds carbohydrates, and dextran happens to fit into its binding pocket. We do have chips that do not have dextran. They're either planar or they have polyethylene glycol on them. Those would be common alternatives when you have something that specifically binds dextran. And then it's not unheard of that you're going to be working with two binding partners, and one of them is just inherently stickier than the other. And usually the way around that is to put that stickier molecule on the chip surface as the ligand and use the less sticky molecule as analyte. That's kind of a quick 30-second run through of what you can do about nonspecific binding. Okay, if you want to identify binding partners of a specific known protein, but we don't know what we're looking for as a binding partner, then it is possible to use this. On the Biacore T200, yes, there is a binding partner recovery mode. You can take an unknown mixture, inject, measure binding, wash the surface. As long as dissociation rate is not very fast, you can then regenerate and recover the bound material, take to a mass spec for identification. There are some papers that have described this in the literature. I won't say it's been a huge Biacore application over the years, but there are probably about between 25 and 50 papers where this has been done and described. How do you convert resonance unit to site occupancy? Because K on, K off would need that. Very good point. I'm going to actually go back a few slides here. There. Here we have Rmax equation. This Rmax is your expected maximum binding signal based on given ligand density, molecular weights of the analyte and the ligand. Now, the Rmax can also be that, it is also that condition where 100% of your ligand is occupied. Any other condition less than Rmax is essentially a measure of occupancy. And if you take that Rmax value, or sorry, that R value and compare it to the Rmax, you will get at any given point in time effectively percent occupancy. And our fitting software and pulling out K on and K off are actually solving for K on, K off, and Rmax in a nonlinear regression. So that's how we are getting those parameters, and hopefully that will be a sufficient explanation. We do go into it in a bit more detail in our kinetics classes that we teach on a regular basis. So let's take a look at Biacore applications. Just give you some examples of how our instruments have been used and how flexible and versatile they can be. So we'll start with binding kinetics with respect to drug design, measuring K on and K off. We have a model system. In this particular model system, we're using thermolysin as our ligand on the chip surface. And we are targeting 17 thermolysin inhibitors. We're closely related, molecular weight around 450 compared to thermolysin molecular weight of 38,000. All of these particular inhibitors are known to bind, but they're all being modified at this P2 position with a variety of side chains to see if you can affect, improve, or modify binding behavior. Now, I will say that small molecule binding to targets is probably the second largest Biacore application now. Over our 30 years of existence, approaching 32 now, it well exceeded 50,000 references in the literature, and probably about 20% of those are small molecule assays. So let's move forward. How do we set this assay up? In this particular example, we're going to use our biotin capture kit. We've got biotinylated thermolysin. We do have streptavidin and neutroavidin coated chips as well. The drawback with those chips is once biotin is captured by streptavidin or neutroavidin, it's effectively irreversible. It's as if we have made a covalent bond. Anything that we do to disrupt that bond denatures the streptavidin, so we can't recapture thermolysin. So we've come up with a very nice reagent. We have a center chip that is coated with a single-stranded DNA. We inject complementary DNA, which is biotinylated, plus streptavidin. We can inject then a biotinylated molecule, capture it. measure binding by injecting binding partner, the analyte, and then regenerate by disrupting the DNA-DNA interaction. Everything washes off. We're ready for another cycle where we will build the surface and measure under different conditions. And we can do this repeatedly. So with that assay format in mind, here we are looking at kinetic rate constants, sensorgram binding profiles. We're doing dose response curves in one sensorgram, proceeding from low to high concentration. And just by visual inspection, you can see there are binders that interact relatively quickly, dissociate relatively quickly. There are binders that might interact more slowly, but also dissociate more slowly, and interactors that are intermediate. Here you can see for series A, 5 compounds with various branched groups here, all of the hydrophobic nature, with group 3, unbranched, but longer, having the longest residence time. Series B, we've thrown on a different branch modification, and we are seeing relatively fast dissociation again. Series C, a long variety of different alkane groups with different optical properties, and most of these had similar K-on, K-off properties. We can actually plot this out. If we make a plot with the log of Ka on the y-axis, log of Kd on the x-axis, you can see there's not a huge difference in K-on over these compounds. We're going from 4.5 to like 5.2 for K-on, but we can get up to 2-fold log shift, 100-fold difference in K-off with relatively little effect of K-on. So these are all molecules that would bind in a similar manner, but then dissociation changes significantly. And this difference in K-off has to be related to the small variations that we're generating at this P2 site on our inhibitors. Doing some structural analysis, combining the Biacore data with structural data, X-ray crystallography, you can see that there's an interaction with asparagine 112 in the active site pocket that is enhanced with this particular series A inhibitor with the branched hydrophobic group here compared to series B where the modification is a little bit different, and series C where there's been a significant change in the binding pocket. And we're proceeding here as the better the interaction is with asparagine 112, the stronger the dissociation rate is, the slower the dissociation rate is. So essentially, drawn conclusion was that combining the crystallography data with Biacore data, that this asparagine 112 is a rate limiting step for dissociation, that it wraps in and locks things in a pocket, and if you lock it in, then it can't dissociate, and that will effectively lead to inhibition of thermolysis. Okay, so now we'll move from a fairly well-defined small molecule protein binding system to a series of more complex interactions with membrane proteins. Membrane proteins are usually, for most biological purposes, more difficult to work with because you have relatively small population of target proteins in a membrane environment and mixed together. You have a variety of different proteins in this membrane environment. It's difficult to purify them out. Can be done, but it usually takes a significant amount of work. If you keep them in the membrane environment without some purification, then you're going to have a relatively low density of targets. And yet about 20% of total proteins are somehow membrane associated, either by passing fully embedded into the membrane or being associated at either the cytoplasmic or the extracellular side. And I think the number is that about 50% of known druggable targets happen to be membrane, transmembrane, or membrane-associated proteins. So they are a very important class of proteins from a pharmaceutical standpoint. So how can we do membrane protein interactions using Biacore? How can we measure these interactions? And there are a variety of approaches. They're published in the literature, working with soluble protein domains, usually extracellular or intracellular domain. We can have done extractions and protein reconstitution in a membrane-like environment. Might be using a VLP or a nanodisk, or working with detergent solubilized protein without reconstitution. This is probably the most common approach. There have even been some examples of working with immobilized cells. And if your binding partners are sufficiently large, protein-protein interactions have been successfully measured with cells as ligands. All right, so here are some examples of membrane protein work. This first example going back about 18 years now. We are looking at receptor purified and then reconstituted on our center chip L1. Center chip L1 has hydrophobic groups attached to the dextran, allows you to capture membranes as either vesicles or intact bilayers, depending on membrane concentration. So essentially, these were receptors with epitope tags captured by antibody and then built the membrane back up around the receptor. Here we are looking at conformational specific antibodies binding to the receptor. All three of them are capable of binding, whereas irrelevant antibodies are not giving us significant binding signals. We can get similar effects by just capturing the receptor on sensor chip L1 or sensor chip CM5, again, without reconstituting lipid around it. So the appropriate detergent conditions can give you binding similar to that of a reconstituted receptor. Of course, here we are looking at fairly large binding partners, antibodies against receptors. That being said, people have successfully moved forward. Here we're looking at CCR5 receptor binding peptides on the 1,000, 2,000 Dalton range, or small molecules of 500 Dalton to solubilized receptors captured in sufficient density on a center chip surface. And these are quite clear, fittable kinetic data. Here's another example of small molecule binding. Instead of a GPCR, here we are looking at a 4 transmembrane segment receptor that's involved in nuclear lipid binding. And here we are looking at 2 different small molecules binding, getting kinetic fits for this flap that has been solubilized and captured in a suitable detergent environment. Yet another example here, we're looking at olfactory receptors, which are again GPCRs, using a specific antibody to capture the appropriate receptor epitope tag in looking at various odorants, binding versus no binding for a nice solubilized receptor. Here is an example of a stabilized receptor. There is a company in England, Heptares, that goes through and modifies GPCRs to make them more thermostable, which then usually makes it easier to find suitable conditions to run assays with solubilized receptor. Here we are looking at a case where the receptor has been captured using our his tag sensor chip NTA. In this case, you did get better results by using what we call a capture cross-linking approach. The capture on NTA alone gave significant drift, but if we got the ligand on chip surface and then used a short pulse of amine coupling reagents to lock it onto the surface, we had much higher activity, although note that you are getting perfectly reasonable data to analyze, even at the lower surface density associated with the capture alone. Again, you can see a relatively wide range of affinities, 7 nanomolar tight binder small molecule to the receptor, all the way to a weak 84 micromolar binder. And another example, this should be wrapping things up. Here we have a full-length membrane associated protein that's been captured through a His tag and then had lipid environment reconstituted around it. This particular example, the protein is expected to function both in endosome environments with low pH and at neutral pH on the cell surface. Here, one particular binding partner, small molecule, works quite nicely in both environments with relatively little change in affinity. Second, larger peptide mimic binds nicely at pH 4.5, but effectively has no binding at 7.4. So see that there's something that's popped up in the chat. Okay, so that, all right. So let's move on. Active concentration measurements. I had mentioned this early in the talk. Why might you want to use Biacore for concentration assays? So essentially, whenever you've got a population of a biological molecule of interest, not all of that biological molecule may be in an active stable conformation. Here we have an example of 20% of our binding molecule being in an active conformation with the suitable binding epitope, other molecules in various states of inactivity or unfoldedness. In general, your classic protein assays, OD280, Bradford assays, Lowry assays, measure total protein concentration. It will tell you that all of this population is present. Biacore concentration assays will focus on the active concentration. And the difference between active material and total material can have a significant impact on the assays that you may want to use. So there's 2 different ways to do concentration measurements in Biacore. One are calibration-based assays. We have a known standard, measure the binding level of that standard at a particular point in time, plot that, compare our samples to our standard curve, and pull out concentrations. Pretty straightforward approach, not necessarily all that different from any other assay that would use a standard curve. The key point here is that we are actually measuring a binding activity to give us our concentrations as opposed to a total protein measurement. By comparison, there is what we call a calibration-free concentration analysis approach, or CFCA. Here, if we take advantage of the flow-based properties of a Biacore instrument under conditions where we have a very high surface density, we can actually get what's called mass transport limited binding. If we're trying to measure kinetics, mass transport limitations are not desirable. We typically would use low surface density for kinetic assays. But for concentration assays, we can actually take advantage of a mass transport limitation. We can inject our sample at 2 different flow rates, which will have 2 different diffusion distances to the chip surface. We're now measuring binding as a function of diffusion rate and concentration. So we'll have 2 different slopes here and can fit our binding level as a function of the hydrodynamics of that protein and total concentration. Here is an example of a concentration assay using a calibration curve and a fairly large binding partner, in fact, a virus. So we've put a receptor for the virus, and this particular virus, Coxsackie adenovirus receptor, or adenovirus, and the receptor is Coxsackie adenovirus receptor, or CAR. So we've put CARs on the chip surface, and we will bind Coxsackie adenovirus. We have immobilized receptor, mobilization conditions, NHS EDC activation of the dextran, passing over the receptor in a relatively low pH solution to electrostatically attract protein to the chip surface. form covalent bonds. Use ethanolamine to block any remaining active sites. And here is our final binding level, up to, in this case, 2,300 resonance units of CAR on the chip surface. Passing adenovirus over the surface, it was necessary to optimize binding. Regeneration conditions had to be worked out that would successfully remove adenovirus without affecting ligand activity. After testing multiple conditions, final results were 2 injections of 10 millimolar glycine pH 1.5, and then 60 seconds of buffer wash afterwards to let the surface stabilize. Going from there, we have a very reproducible assay. We can use a virus standard from ATCC to give us a standard curve, and the range can be from 0.11 to 14 viral particles per mil. So we're diluting our typical samples 100 or 200 fold to reduce the final total number of particles in the screen. But the assay is quite linear and covers a relatively broad range of 10 to the 9th viral particles per mil. Yeah, you see the exponentials did not transfer very well on this slide. I'll go back and fix that. All right, so hopefully what I've managed to convey in the last 45 minutes or so is that Biacore T200s are very sensitive, versatile, and efficient instruments, giving you a wide range of biological interactions that you can measure, all the way from small molecules binding to protein or other biological molecule targets, up to cells and viruses interacting with biological molecule targets, and is a very reliable and reproducible instrument as well. And at this point, I'll be more than happy to take any questions that you may have. All right, so we've had some things pop up in the chat. Let's take a look. All right, so, okay, are coated chips with the advantage of regeneration as durable as non-regenerated chips? So the single-stranded DNA chip is very robust. Those chips can last for months because DNA is, as far as biological molecules go, a very robust surface. We can see similar performance with chips that are coated with IgG. Antibody, IgG in particular, tends to be a very robust antibody. You can make a surface between use, store it in the refrigerator, 4 degrees C in a moist environment, and expect to use that chip for 3 to 6 months. Other proteins are more labile. Protein A and protein G, which we do sell chips coated with both protein A and protein G. We recommend immersion of those chips in buffer and store at 4 degrees between use, and they will usually be good for up to 3 months under those conditions. Other ligands, I once did some experiments with IgE. Initially, the thought was that IgE would be very similar in stability to IgG. Turns out it's not. It's much more labile. We ended up making a new sensor chip a new sensor chip surface every day. We would make a chip in the morning, run their experiment overnight, next day, start again. So it really depends on what you're putting on the chip surface. Molecules that are more stable, you may get 1,000 binding cycles over a 6-month period. Other surfaces may only last for a couple of days, which brings me to the advantage of epitope tags. If you can engineer in an epitope tag to one of your binding partners and capture through that epitope tag, now your chip lifetime becomes the lifetime of the molecule that you use to capture the epitope tag and not the ligand itself. Okay, let's see, there are further questions. Enzymatic reactions with small ligands where there are covalent bonds broken and formed. There have been limited successful attempts to measure enzymatic properties through SPR. And effectively, all of the assay formats require some sort of mass change happening to the ligand, that the enzymatic activity either trims the ligand or adds mass to the ligand. So that gives you a visible property change. Otherwise, if you're just changing the analyte, you'll see a binding event for the analyte, and you'll see dissociation of analyte, whether or not it has been catalytically converted. And so you're not going to be successful in general measuring catalytic events on analytes. But if you can put a ligand on the surface in a reversible manner and measure mass changes to that ligand, then you can do classic enzymatic reactivities and measure things like kcat. Next question. Plots the baseline after dissociation. For most of the plots we showed, we came close to baseline, I would say. But yes, and ideally, we should be coming back to baseline completely. In a capture assay where we are washing ligand off the surface, we may have to adjust for changes in ligand density as the assay goes on. We might not capture as much ligand at the end of the assay as we did at the beginning of the assay. But there are ways to adjust for that. Of course, optimizing regeneration conditions to get baseline as close as possible, or base signal post-regeneration as close as possible to the original baseline, is the optimal way to go. Okay, if we're getting significant non-specific binding, that probably has to be dealt with first. That we need to minimize that non-specific binding before worrying about other aspects such as optimal regeneration. Now, I will note that I have observed on occasion a regeneration condition that does not appear to fully remove bound analyte from the chip surface. But then when you go back and run a replicate sample, you have the same binding level, the same kinetic parameters, same Rmax, or binding level for that particular concentration. And under those circumstances, I tend to attribute that to a denaturation / precipitation event for some percentage of the analyte, that it is coming off the ligand, but it's not necessarily clearing the center chip surface. So if you are running a well-designed experiment, you will have replicates, and those replicates will give you the same binding level at the beginning and at the end of the assay. And hopefully that helps. What can you do to damage an L1 chip? L1 chips are sensitive to being left out to dry. Exposing them to air for extended amounts of time can lead to oxidation of the hydrophobic groups. Additionally, if you pull the L1 chip out of the instrument without washing the lipid off first, then the lipids will also dry, and you can't really get them off the L1 chip once they've been baked on in that manner. So those would be things that I would be aware of. I typically use a strong detergent like SDS at the end of the assay to wash all lipids off, and then store the L1 chip actually in buffer or water between uses. Okay, binding events that involve change of conformation. That's an interesting question. If the conformational change is occurring on the time scale of minutes or longer, then there's a chance that it can be seen in a Biacore assay. And usually this will be noticed as a change in off rate as a function of contact time. That if you are moving from an initial docking state to a tighter final conformation, then if you inject your analyte for 1 minute, most of your material is still in the initial docking state. You inject analyte for 5 minutes at the same concentration, you see a shift towards a slower dissociation rate as a function of contact time. Mind you, there are some people that think that this is usually some other sort of artifact and are not eager to tie it to conformational change. But if you can validate the data that you're seeing in the Biacore with other assay formats that also show conformational change, you're usually standing on more solid ground. Okay. Chips designed to immobilize carbohydrates or maltose binding protein. My experience with maltose binding protein is that it's actually a relatively weak epitope tag. It dissociates very quickly from maltose, so you don't get stable surfaces. For immobilization of carbohydrates, usually aldehyde-based coupling can work, or synthesis of a carbohydrate with a biotin group somewhere in it. Those are probably the two most common approaches for putting carbohydrates on sensor chip surfaces. Okay. How long does a histidine tag protein coated on NTA chips? Well, the whole purpose, it depends on what you're trying to do. Let me rephrase that. The advantage to a capture system is that you can wash off ligand every cycle, and so you don't have to worry about the effects of lifetime, stability, or regeneration solutions on the activity of that ligand. That being said, if you've got a stable molecule or you need a high surface density, you can use that capture cross-linking approach that I mentioned earlier to give you an oriented epitope tag molecule that you do not have to replace on a cycle per cycle basis. If your dissociation rate is relatively fast, as it may be for small molecule binding, then you might skip the his tag, the EDTA buffer to wash his tag protein and nickel off the NTA chip. That can also be a valid approach. However, you do have to have a fast dissociation rate on the order of 10 to the minus 2 per second or faster. At rates significantly slower than 10 to the minus 2 per second, it's going to take hours for your analyte to completely dissociate, and that's usually not practical. Okay, do I have chips to immobilize carbohydrates or MBP tag? Yes. DNA-coded chips with specific sequence of our interest? Unfortunately, no. Our biotin capture kit has a specific proprietary, unknown to me sequence, and that's what we use. However, there's nothing stopping you from using a biotin tag to put your own specific DNA sequence on a streptavidin or nutravidin chip. If you have a single-stranded DNA, then you could then capture a second complementary DNA with its own tag. Or there are ways to amine couple DNA to our sensor chip surface. That's essentially how we are making our biotin capture kit, the cap chip. So you can do amine coupling of DNA to put your single-stranded DNA on the chip surface and then use a similar approach depending on your assay goals. Okay. Yes, CM5 chips can definitely be used. Our instruments can tolerate up to 10% DMSO. Other common co-solvents may be at lower concentrations. We do have a chemical compatibility guide in the manual for our instruments. L1 is probably less optimal for organic solvents because you're typically using the L1 chip to capture membranes. And those membranes are going to commonly not be stable in the presence of those organic solvents. Yes, lipid vesicles can be immobilized on chip L1. And are there other questions? That seems to be as far as I can scroll at the moment. Well, I see one question came in the chat, and I just posted my email address. If any of you have questions about design of Biacore experiments or interpretation of Biacore data, please feel free to reach out to me. I cover all of the central US. So even if you're not based in Indiana at the moment and are sitting in on this discussion, please feel free to reach out to me about your Biacore experiments. Is there a specific lipid composition that needs to be used with presumably L1 chips? No. In fact, it works with most liposomes that have been prepared. What you may see is, depending on lipid composition, a difference in whether you're capturing your lipids as intact vesicles or as a planar bilayer. And typically how you would tell the difference is that intact vesicles will have about twice the total mass of a planar bilayer. L1 chip can capture usually up to 8,000 RU of a vesicle, 4,000 RU of a planar bilayer.
Okay, I think with that, we'll call it a day. I want to thank you both again for participating in the seminar series, and hope you have a good day. Thank you, everyone.
Thank you.
Thank you.